said, most of the digestion in humans occurs in the small intestine. Most of the nutrient absorption also occurs in the small intestine. And there are three modifications that facilitate that. But one you can actually see with the naked eye. And these are circular folds. And you can see them right very nicely here inside the duodenum, like little rings here. These are called plicae circularis. Plicae circularis, these circular folds, you can see with the naked eye, in the mucosa and submucosa of the small intestine. Now these are permanent folds. Unlike the rugae of the stomach, which flatten out as the stomach distends when they enter food, these are permanent. So what they do is they help to slow down the passage of material, you have more time to work on it, and they also give you more surface area for nutrient absorption. Just like in our lungs, we have all these tiny little air sacs, alveoli, give more surface area for gas exchange. Here with all the folds, you're going to have more surface area for nutrient absorption. So that's the first modification. Macroscopic, you can see it. Folds in mucosa and submucosa, plicae circularis. The second one you can't see except under a microscope. And these are little finger-like folds, okay, and you can see that 876, 879 in the mucosa of the small intestine. They're called the villi. So these are folds in the mucosa of the small intestine, which further increase the surface area for absorption. So I got these big folds, plicae circularis, and you got the small folds, they're finger-like extensions of the mucosa. Within each villus, that's the singular villi, there is a blood capillary network to absorb nutrients, and then a lacteal, shown in green. What's a lacteal? Anybody remember? It's a lymphatic capillary. Why do you want a lymphatic capillary there? There's two of you to ask that question. You guys know? Yes, lipid absorption. You say, why don't they just get absorbed in the lymph nodes? It's a very curious habit. It's digesting our lipids, particular fats, break down glycerol and fatty acids, and they pass through the epithelial cells that line the mucosa. They get reassembled as fats. Now they're too big to get through the walls of the blood capillaries, so they're absorbed by the more porous lacteals. And that's why your liver is full of fats. It goes right to the blood, and you digest those fats in the blood. That's why after a big heavy meal, you take a blood sample, it looks like cream. Look much like blood at all. That's why too they're saying if you have a blood sample drawn, fast beforehand. You know what they're doing? Maybe 12 or 6 or 8 hours. Well, you expect it's just full of fats, lipids, and so on. In your meal. Because where does the lymph go? Back in the blood. So you're dumping all those fats back in the blood, you're going to redigest it in your blood. Okay, so that's two modifications. The villi, and you see the plicae circularis, which you can see here, and the villi, which are microscopic, you see in the microscope, and I'll show you those on Thursday. The third are actually tiny little folds in the cytoplasmic membrane and the epithelial cells that cover the villi. And we can't see them even in a light microscope. They're too small. We require an electron microscope. So what we will see, it looks like a fuzzy border. So when you first look at it, it looks like, hey, it's ciliated. It's not ciliated. Those are actually microvilli. We just can't see them clearly with a magnification light microscope. And this further increases surface area for absorption. And we're going to see there's important enzymes, digestive enzymes, that adhere to this. And by the way, this is called the brush border. It looks like a fine bristled brush. These are actually microvilli, not cilia. Because it looks like a brush, it's called a brush border. We have some enzymes that adhere to those, called the brush border enzymes, that complete the digestive process. We'll talk about this in lecture. So those are three modifications. Three, not four. Right? Plicae circularis, villi, and microvilli. All these serve to give more surface area for absorption of nutrients in the small intestine. Okay, Peyer's patches. We talked about that before. Peyer's patch is part of the malt. So these are collections of lymphocytes in the wall of the small intestine. In the mucosa and sometimes down in the submucosa. Malt, mucosa associated lymphatic tissue. In the small intestine and the fancy name called Peyer's patches. Intestinal crypts, also called crypts of Leverkuhn. These are actually tiny little tubular indentations at the base between adjacent villi. intestinal crypts or crypts of Leda too. The little tubular pits at the base between adjacent villi. You see in the small intestine as well. Okay, then there's a sphincter between the ileum of the small intestine, the distal part, and the proximal part of the large intestine, which is called the cecum. So it's called the ileocecal sphincter. And this regulates passage of materials from small into large intestine. As you can see here in this model, it's constricted area right here. Here's the ileum of the small intestine, and here's the cecum of the large intestine. So this sphincter right here is called the ileocecal sphincter. Okay? The ileum of the small intestine, cecum of the large intestine. It's constricted area right here. Again, that radius passage of materials from small to large intestine. Again, it's another valve. So a little bit passage from small intestine and large intestine. This regulates the passage. Ileocecal sphincter. Okay, the large intestine. You turn to page 888, 891, your textbook. The diagram of the large intestine. Again, I'd like to know the regions of large intestine. So if you're doing a trace, you can tell me the parts. The proximal and distal. We start with this little pouch region. This is the proximal part of the large intestine. And it's called the cecum. Now in humans, it's not very well developed. In animals that eat a lot of plant material, it's usually much better developed. You'll see it's better developed in a pig, for example, because this usually houses microorganisms that produce enzymes to break down things like cellulose in cell walls of plants these animals consume. Cellulose is a complex carbohydrate made up of many molecules of what? Remember the name P1? Sugar, right? Sugar. Glucose, which is a perfectly good food molecule. In fact, our brain cells need it. Glucose, that's your blood sugar. Okay?